Hi. I'm glad you guys are still here because it's getting late, I know. I'm going to talk today about some trends in early plant-pollinator community recovery following hurricanes. The data I'm going to present today, first of all, is only from San Salvador, although Nancy and I have worked on several other islands. We have a huge data set. We've been working together on this general project of plants and their pollinators, the networks, for about 6 years now. Let me turn this a little bit. That's a little better. And today what I'm going to present is just data that we've collected in Decembers since 2010. Okay, so I know a number of people have addressed issues around the ecological and economic importance of coastal communities in the Bahamas. I love to hear that because I hate it when I'm the only one saying it. Ecology and economy are definitely tightly tied together, especially when you're talking about coastal communities, because ecologically, plants keep erosion down, they stabilize dunes, and economically, since the tourists prefer to be on the coastline, they also, while many of them are not here to look at plants or pollinators, they do realize when the plants are gone. And so it's really important economically as well. I'll give you an example. So these are two different beaches. The one on your right-hand side is East Beach following Hurricane Irene. So it was about 3 months after the hurricane. And you can see that there's, right in here, there's a deep drop. That's about a 2-foot drop where the foredune eroded away. This other image is following Joaquin. This is down near the mouth of Pigeon Creek, which is the only lagoon system on San Salvador. And you can see dead trees. You see nothing green. This was in December, so 2 months after the hurricane, and it hadn't come back very well yet. But look what happens after a very short period of time. Right, this is East Beach about a year after Irene. And what's happened is not only do you have plants that are now growing where there didn't used to be plants, this is that vegetation line that you saw, but you can see that there's also sargassum and other, probably some turtle grass and things like that that have washed up, and those actually build substrate for the plants. We think they provide both nutrients and anchorage. And so together, you end up with pretty plants, which make pretty flowers, and from our perspective, bring pretty bugs. Now, if I can only move forward. Okay, so I know I flipped twice, that's okay. We have, Nancy and I firmly believe that we should be using nature as a model for sustainable conservation. And the reason is because nature does it on its own. What I'm showing you here on the left-hand side, this is a typical foredune beach community. You got a deep drop, that's because it was following a hurricane. Over time, if deposition was occurring at this location, you would end up with the foredune becoming much bigger, and behind it you might find communities like this Cocothrinax shrub community. Now, if it's really windy all of the time, it tends to stay this way. But if not, you'll end up with shrub thicket right here. Very thick, as others have mentioned, hard to walk through, especially if there's a lot of Mimosa bahamensis. And over additional time without disturbance, you end up with coastal coppice. That's a young coppice right there. That's me. I'm not very tall, right? So nature knows how to do this. And if we can understand how that happens, what processes are necessary, what species maybe are critically important, then we can model that. And by modeling that, perhaps we can be more efficient with our limited funds for conservation efforts. Now, Nancy and I have been working on, this thing is bugging me, Nancy and I have been working on the basic premise that communities are built on their interactions. Okay? So we have been focusing primarily for the last 6 years on plants and their pollinators. Here's a couple of nice examples. This is one of our favorite bees, Megachile alleni, sitting on Cocorus hirsutus. And for Nancy, here's a wasp. She's a Hymenopterist. That's Campsomerus trifasciata, and it's sitting on Jackmontia. I think it's Jackmontia kansas. And the reason that we're interested in plants along with the pollinators is because if you only want to conserve plants, and that's what people see, people who are not biologists, they see the plants, they really don't want the stinging insects. But without those insects, you're not going to maintain viable populations of the plants. And so it's critical that we think about both of them. And technically speaking, pollinators need a lot more than just floral resources, right? They need the floral resources, don't get me wrong, especially if you're a bee. Bees require pollen for the babies, always, and nectar for the babies and themselves. But there are more than just pollen resources and nectar resources that are important even for bees. And if you're talking about other kinds of insects, we have other issues as well. So, sometimes we need other kinds of food, right? In addition to, if you're not talking about bees, we got a lot of syrphid flies, we have lepidopterans, we have other wasps, and some of these wasps are good pollinators. They're carrying pollen from place to place. And so we need to make sure that those food sources are also available for the pollinators. They need nesting materials. This is a picture of a Megachile alleni nest cell. They are ground nesting under rocks. We lifted about, I don't know, myself and a couple of students lifted about 10,000 rocks to find about 200 nest cells. And they are wrapped in leaves. It's a leafcutter bee. And so we need to consider the nesting materials, the availability of nesting sites, as well as that other food if we're going to conserve the pollinators, which are critical for the plants. Now, because we're working in the Bahamas, and that's intentional, no question, one of the major disturbance events is hurricane disturbance. And so we ask ourselves a lot, can hurricanes have positive effects on diversity? We know that hurricanes can be devastating. They can rip branches off of trees, they can wipe a community clean, right? But we think they can also provide positive effects. For example, they bring resources. In a dry location, a lot of rain, a lot of fresh water comes in. That is a resource that is limiting often for these insects and for the plants. Detritus, all of the dead stuff, mineral nutrients and food for other organisms. And it opens up space in the community. So hurricanes do have a lot of negative effects, but they also have positive effects. And those positive effects could be really important. Now I'll get to the point. That was my spiel for why you should care. Why should you care? Because insects are important. To understand the purpose of the talk, and what I'll talk about the rest of the time here, is to understand recovery of these networks of plants and pollinators that occur after hurricanes. And specifically, we want to address things like which plant and pollinator species are active very quickly following hurricanes. So which plants are in flower, which pollinators have mature individuals flying around in the community. We want to know if generalists are more likely to recover quickly than specialists. That's important, both from the plant and the insect perspective. And in terms of the pollinators themselves, we want to know if their nesting requirements influence their recovery. Because, if it's a ground nesting insect that nests under cocoa bushes on the edge of the beach, and you get a big surge, what kind of an impact does that have on those developing larvae versus something that's nesting by burrowing into a tree? So, most of the work that we've done, like I said, all the work I'm going to present here is from San Salvador Island, and I won't go into any more detail about that. We have worked on other places. I also mentioned that we're only presenting data from December. And these are the 5 years that we've collected this kind of data, 2010 through 13, as well as 2015. It can tell you about 2014 some other time if you want to know. We also have similar data collected in March and June of every year since 2010, as well as several years of late July and 1 October, where we spent time down here. In each of these December trips, we spent between 5 and 9 days doing data collection for a total of 30 to 60 hours in each of those trips, depending on the trip. The total amount of time we spent making observations thus far is 240 hours. What we did while we were out there was record all of the interactions that we saw. Those would be the floral interactions between the pollinator and the plant, as well as any additional pollinator activity, because as you know, sometimes insects do not land where you would like them to land. Instead, they flitter about, and you wish they'd land so you'd know what they were going to do. Now, we collected data in 3 different ways. We used a narrowly defined watch zone where we also estimated floral intensity, so how many flowers there were in a square meter. The purpose of doing that is so we could also estimate visitation rates for each of the species. In addition to that, we collected along transects, usually 20 to 30 meters long, sort of a walking transect. You walk back and forth, and whatever insect you see, you record, and that way you can actually follow them and see if they are loyal to the same species or if they are bouncing between plant species. And then finally, we collect insect vouchers, and each one of them we remove their pollen load and identify the pollen to species when we can, and genus more often probably, which gives us trace evidence of where they've been even if we didn't see them. So using these 3 methods, we're hoping we're getting a good picture of which insects are visiting which plants. In addition to that, for the last 3 years, we have been marking individual pollinators now to estimate their population sizes, at least in terms of the mature individuals. As you are probably aware, insects are rather cryptic. Before they are reproductively mature, they're hiding from us. It's really difficult to measure their population sizes outside of mature individuals. And so what we've been doing is marking those mature individuals with tiny dots of Tester's enamel paint of varying colors on the dorsal surface of the thorax. And as long as you don't hit the wings, everything is fine. Here's a couple of examples. This is a male Campsomerus trifasciata. It's got a tiny little red dot. Oops, wrong button. Right there. Another example for you is Palpada albifrons. This is a syrphid fly, and it has a purple dot. It's wearing purple. And in case you're wondering whether or not this has an influence on the organism's ability to go about its work, as long as you don't hit the wings, it doesn't seem to have an effect. I have one really nice picture here to show you. This is Megachile alleni. It's a leafcutter bee. There's its green dot on the thorax, and it is carrying a leaf fragment into its nest, which is located between these 2 chunks of rock. So they're doing what they ought to be doing despite carrying paint on their backs. Now, during the time frame that we've been studying, so that's since 2010, there have been 3 hurricanes that have hit San Salvador Island. We had Irene in 2011. That was an August storm, category 2 to 3, depending on which day you looked or what time of day. Sandy in the Bahamas was category 1 to 2, and that was October of 2012. And of course, Joaquin, just this last year in October, was a category 4 for a very long time. San Salvador is in the bullseye there in each one of those cases, so you get a sense of where we were. Right? So, let me show you what we've got here in terms of results. I want to let you know, first of all, that plant damage was consistent and correlated with the strength of hurricanes. Okay, so the weaker storms, Sandy, followed by Irene, did not have nearly the damage to the plant community as Joaquin in terms of broken branches, etc. In fact, when we came back after Sandy, it didn't really look any different than it had in the previous June, right? Other than a bright flush green leaves. On this graph, what you're going to see here, I've got 2 of these. Hurricanes are up here. So these are hurricane years. I've got plant families and plant genre in each of those years, the number of plant families and plant genre. We have about 1,500 separate observations that we're trying to process, so this is a very rough estimate of what's going on, our preliminary results. You can see that there is not much difference, actually, in terms of the number of plant families as you move through time, although there is a drop between 2010 and 11 versus 2012-13, and then Joaquin actually did take a hit, right? The community took a hit. Similarly, you see that we have a decline after Irene, and then it sort of plateaus. It's the same number of genera here. And then we have another steep decline, and that's, in both cases, the difference between 2010 and Joaquin is about 2/3 the number of families and 2/3 the number of genera that we saw active. Active in plants means they have flowers. Now, I will point out to you that prior to 2010, the last hurricane that came through on San Salvador was Francis in 2004. So this community in 2010 had 6 years to recover. And in the last 5 years, there have been 3 hurricanes. So we aren't going to see the same kind of recovery all the way back to what we have in 2010 until we got a length of time that would be appropriate. Now, here are the insects. And again, it seems a little slushy, I know. It's interesting, isn't it? The kinds of effects. We've got a decline and then a slight uptick in terms of the number of insect families represented, and then it drops back down here in 2015 after Joaquin. And again, we've got this sort of drop and a plateau and a drop again in terms of the number of genera. In both cases, both in terms of the plants and the pollinators, these are not the same genre. We have different genera that are more prevalent, and if we look at the relative frequency of visitation among these different genera, we see some really interesting patterns. And actually, that's what I'm going to finish up with here, is to share with you a few patterns for what I like to think of as a select group of species, and they're not all species that make sense, just to make you know that I'm not doing that. So here we've got Campsomerus traphaceata. In 2010, we had a lot of individuals observed, dropped precipitously. In fact, we were worried about them. These nest under, as far as we know, they nest under Cocoloba, and they are going after beetle larvae, which are found under those Cocoloba. And so we were concerned about them because of surge effects. Megachile alleni is a real workhorse. It is active year-round. Every time we're here, we find mature adults. And as you can see, we had a lot of individuals early on dropped off, and this year we saw none. 0. Scary from our perspective. Xylocopa cubaecola has an interesting pattern. Dropped following Irene, but then climbed back up and doesn't seem to really particularly have much of an influence, this Joaquin storm. Look at Palpada. Palpada is really interesting. This is a dirty water nesting fly. After big hurricanes, their population sizes increased dramatically. And then in terms of lepidopterans, I've got 2 to show you. In both cases, we've got declines following hurricanes. Agralis, which is the Gulf frit, did not really come back after Irene. But look at Leptodes, tiny little thing, goes up and down with the hurricanes, right? The issues here that we're facing is for most of the insect visitors that we see, there is absolutely nothing known about their nesting requirements. Very few insects do we know about. That's a big problem for us, and that's where we're headed with this kind of work. I'm going to stop there. I believe I have time to say thank you to these people and probably not time for questions.